Quiz 5. The average grade for that was 61%. The high was 100%, and the low was 30%. Now, on quiz 5, those who got 90% or greater, there were 4. 4 students in this class got 90% or greater on quiz 5. Those who got 60% or lower were 16. So that's for quiz 5. Then I looked at the course totals to date and asked how many got greater than 90% average to date. So this includes all the assignments that were graded. And the number is 3. And if I asked how many got 60% or lower, the answer is 13. And that includes the worksheets. So in the worksheets, since you can look up the answers, the grades are very high. So it's the test which are closed book, that's dragging the scores down. So what this means is that a significant number of students in this class really aren't learning the materials. And the question is, why? Am I not presenting them properly? Are the students not studying enough? I don't know the answer. But I just want you to know that these are the statistics, and I'm doing whatever I can to try to help you out, including giving you the answers and telling you where the questions came from so that you'll know for the next test how to study. You study the slides. So any questions or comments about what I've said so far? Any thoughts about how scores can be improved?
Teacher, I have a question about the time for quizzes because we have less time. Because of this, I cannot concentrate correctly. When the test is finished and quiz is finished, I know the answer. That's my big fault. I don't know about the time. Time is very restrict.
Well, I sent you a memo giving you the rationale for the timing of tests and quizzes. And for most of the people, in fact, all of the people who get good grades, they seem to know the answers. They've studied in advance. They know the material. It doesn't take them a long time to answer the questions. So what I'm encouraging you to do is know the material before the test so that you don't have to think long and hard about it during the test. Okay, anything else? Having said all that and hoping for better going forward, I'd like to go over quiz 5 with you.
Professor, would I be able to say something quick about workshop?
Yes.
Thank you. So, you guys know I have a workshop Monday and Wednesday. It has been asked that I consider adding a workshop hour on Fridays. Unfortunately, that is not, I can't just add an hour, but what I can do is move one of the existing workshops to a Friday. So I put something up in the discussion thread, basically a proposal. There are 2 reply threads that I started, a yes or a no thread. What I would like people to do to gauge interest is go to that discussion thread and reply under either the yes or the no. So we can just sort of total things up and see what people feel would be their usefulness, the usefulness to them for moving a workshop from either Monday or Wednesday to Friday. And if there's a big enough response, then we can go ahead and do that. But due to restrictions from the school, I don't really have an option to add any more workshop hours.
Okay, everybody respond to Scott and see if more of you can attend workshops and get additional instruction on these materials. Can everybody see the screen? Quiz 5.
Yes.
Yes.
Okay. All right. Going over the questions and have you tell me what the answers are and say your name before you do so that I can record you as having participated. Number 1, fill in the blanks. Basic structure of DNA is commonly described as a what?
Yeah,
Stranded, double what?
Yeah, double strand and double.
Double stranded, double what?
Helix.
Yeah, okay. That's correct. And that here's the slide that question came from. The discovery of the DNA molecule was momentous. The scientists who discovered it, Watson, Crick, and Franklin, were all awarded the Nobel Prize in 1962. True or false?
False.
Okay, it is false. And why is it false? Remember
I know.
Okay, Scott, you don't count.
Zach Nussler, isn't it because Rosalind didn't get the Nobel Prize?
She did not. And there's a lot of criticism. And this was in the 1950s. And women didn't get a lot of credit for doing science in the 1950s. And there's a lot of criticism now that women are more aware that she was really cheated because it was her picture, really, that allowed Watson and Crick to figure out the structure. So anyway, that's false because Rosalind Franklin was not included in the Nobel Prize. Number 3.
The actual reason she didn't get it is because she died of cancer in 1958, and they will not award a Nobel Prize to somebody who's deceased.
Yeah, I can't remember. Yeah, 62 was when the prize was awarded. Okay, number 3, we've studied several types of RNA, mRNA, tRNA, and rRNA, and what other type of RNA?
Ribozymes.
Yes, ribozymes. And what are ribozymes known as?
It's a vacuum.
And this is a slide that question came from. It says right at the bottom, ribozymes are distinguished by having catalytic activity.
Yeah.
Other types of RNA are not catalysts. Okay, the next question. More people, different people who haven't responded, please do. It's an essay question. The CFTR protein has 2 nucleotide binding domains. What is the function of these regions with respect to the protein's function? What's the function of the nucleotide binding domains? And here's the slide it was taken from. Some of you got this right, so speak up.
I've already answered.
Okay, somebody who hasn't answered, who got this right, tell us the answer.
It's transportation, teacher.
Okay, Zach, you've already answered, but do it again. Tell us what.
The nucleotide binding domains are there to accept the energy given off when the triphosphate is cleaved.
Yeah, I accept a number of different answers for this, but anything that talked about providing energy to transport the chloride ions, I accepted. The cleavage of the triphosphate to a diphosphate, which is it's a high energy bond, and that provides energy for the transport to occur. Okay, the next one. Terms leading strand and lagging strand refer to what process from that list?
Replication?
I didn't hear that.
Replication.
Okay, that's Mary.
Yeah.
Yes, you're right. It is replication. And that takes us to the next question. A mutation is defined as any change in the amino acid sequence of a protein.
It's
Someone knew.
False.
It's true.
Aisha?
False.
I
False.
put, yeah.
So I've heard both true and false. Someone who thinks it's false, say why.
I think it's true because of the sequence of protein, change the amino acid sequence of protein. It's called mutation.
That is false because it's a change in the nucleotide sequence of DNA as opposed to the amino acid sequence of a protein.
Is that Brian Yee? Okay. Yeah, the slide that the question was taken from says definition. Anytime you see the word definition, memorize that. It's a change in the nucleotide sequence that could result in the change in the amino acid sequence. But by definition, it's not a change in the amino acid sequence. It's nucleotide. Okay, number 7. Which of the following terms is not associated with Okazaki fragments?
Continuous.
Continuous.
Yes, it is continuous.
Because it's discontinuous.
Yeah.
Continuous is not associated, right? Number 8, exposure of DNA to UV causes a thymine dimer, which produces a kink. What's the effect of this on protein synthesis? Someone knew.
This is Marcus, professor. I think when you have a kink, it can lead to mutations in the DNA that will, I think one of them is like a copying error.
Yeah, that's right on the nose. It is likely to cause a copying error, which would be a mutation. And here's the slide for that. Number 9, tRNA molecules function to shuttle amino acids to the ribosome. How many different types of tRNA molecules would a human need to synthesize all the required proteins? Someone who has.
It's
20.
C20. This is
20.
Casey Bostock.
Give me your name.
Casey Bostock. And I
Bostock.
put 20.
20. That's correct. It is 20. 1 for each amino acid. Each amino acid has its unique transfer RNA. Here's a slide that says so. And number 10. Most of you did not get this, which surprised me. So Someone tell me what type of mutation is this and why?
So it's delete. I confused in this question. Instead of I say the type, I said the effect. So I wrote frame shift. I saw there is a missing deleted like amino acid, but I didn't write the type. I wrote the effect.
Well, if you said frame shift, tell me why it's not a frame shift.
Because that's the effect, the frame shift, it's the effect, that's what will happen, but it's
is-
delete.
Is there a frame shift in this mutation?
Yeah, we lose, we have all the chain, it's okay, just we don't have the GGA.
Right, we don't have the GGA.
So I have to say delete, but I said frame shift, the effect, not the type.
Yes, you just said the right word. It's a deletion, not a frame shift.
Yes.
If it were a frame shift, all the sequence downstream of the mutation would be changed. But it's not changed, it's identical. The only thing that's different here is that the GGA is missing in the mutation. So this is a deletion.
Yes.
Not a frame shift or insertion or translocation. It's none of the other things mentioned on this slide. It is a deletion. So make sure you know this. I'm going to ask you this question again in a different way, but make sure you know how to identify what type of mutation has occurred.
Yes.
Anything else about this quiz, the questions, the answers, where the questions came from?
Yeah, I have a quick question for number 5.
Number 5.
So I had answered duplication, but the answer is replication. I'm just a little shaky on what the differences are between the two, because they both seem to imply that the DNA is getting copied.
They do imply that. The term, the standard term for DNA copying is replication, not duplication. So you would never find that, say, for example, in a textbook.
Gotcha. Okay.
Okay, anything else? All right, then I'm going to stop sharing that. Do you see membranes and transport?
Yes.
Okay. So this is the topic for today. What is a membrane? A selective barrier that allows some things to pass through, but stops others. What is transport? The movement of an object from point A to point B. The plasma membrane is a phospholipid bilayer that also contains proteins, and it forms a barrier between a cell's contents and the exterior of a cell. And the picture here on the right is a transmission electron micrograph of 2 cells, cell 1 and cell 2. So you see 2 plasma membranes and an intercellular space between the 2 membranes. And if you look carefully, you can even see the 2 phospholipid bilayers separated by a small space. So the magnification here is very high so that you can actually see what a plasma membrane looks like. A membrane functions. It organizes cellular functions, controls the exchange of materials, and communication and signal transduction, cell-to-cell recognition, transformation of energy, and cell adhesion. These are all functions of the plasma membrane. So what are the components that make up the plasma membrane in addition to the phospholipid bilayer? So there are four major classes of biological macromolecules. The plasma membrane contains three of them. Question to you is, which one is missing? You can think about that when we go over the lipids. Phospholipids, glycolipids, and glycoproteins, and steroids, they're all part of the plasma membrane. There are proteins and carbohydrates. So what major macromolecule is not found in plasma membranes?
Nucleic acid.
Nucleic acids, yes. The other thing I want to point out here is that the proteins and carbohydrates can form what's called a glycocalyx, also known as an extracellular matrix. So let's go into some detail about the glycocalyx. Structure, it's a thick, ultra-sticky, gel-like covering of the plasma membrane. It's composed of sugar and protein strands bound together. So you can see a picture of a glycocalyx here on the right. This is an actual micrograph. And below it, you can see it represented as a diagram. These long chains of purple and yellow are chains of amino acids and sugars that get all tangled up and combine to form this matrix. And here on the lower left, you can see a cross-section of a cell. The surface looks hairy, and that's due to the glycocalyx. So what are some of the functions of the surface layer? Well, stickiness. If you ever touched a cell in culture, you'll know that your finger comes away with gooey stuff on it. That's due to the glycocalyx. Cell hydration to avoid drying, for example, the cornea of the eye. Those of you who like to fish, you try to pick up a fish, it slips out of your hands. That's due to the glycocalyx on the fish scales. And red blood cell volumes are affected by it, and it's a bacterial shield against the immune system if the bacterium is trying to attack. This is a reiteration of what I've just said. Animal cells don't have cell walls, but the glycocalyx gives them structural integrity. That's the main point I'm trying to make here, is that the extracellular matrix, it has a number of functions, but for animal cells, it's for strength as well. The main component of the plasma membrane is this phospholipid bilayer. It's constructed of phospholipids. There are two layers. The outside is hydrophilic, the inside is hydrophobic. So if you look at the picture, we've gone over this before. The phospholipid molecule has a nonpolar tail, a long chain hydrocarbon chain, which is nonpolar. And then it has a polar head composed of a phosphate group and another polar molecule. So the tails, the hydrophobic tails, attract each other. And that's what makes the bilayer. The hydrophilic heads point into aqueous environments. So the inside of the cell is watery cytosol, and cells are always in a watery extracellular fluid. So what the plasma membrane does is separate the cell contents from the rest of the world. The fatty acids, if you look at lower left, the phospholipids can be entirely saturated, which makes for a rigid molecule, or they can be partially unsaturated, which allows them to bend, and that makes for a more flexible membrane. And that becomes important when we talk about fluidity. If you see here in the upper right, a saturated molecule is a hydrocarbon chain with no double bonds. If there's one or more double bonds, it's unsaturated, and that allows the phospholipid tails to bend. Here's a word you need to know: amphipathic. Phospholipids are amphipathic. That means that they're hydrophilic, that the molecule is hydrophilic on one end and hydrophobic on the other. And that's simply the same picture is shown here again to emphasize the word amphipathic. Now, where have we seen that before? Phospholipids are amphipathic because of the function of the plasma membranes. Remember when we talked about the pancreas and the gallbladder? We talked about bile acids. Cholic acid, glycolic acid is an amphipathic molecule. It's a steroid on one end, which is very nonpolar, and it's amino acid on the other end, which is polar. So by definition, it's amphipathic. And what's the function of glycolic acid? It's to digest fats. It has two roles: to emulsify fats and then to solubilize them so that they can be acted on in an aqueous environment by enzymes. So I mentioned this simply so you can see that amphipathic molecules are involved in a lot of different functions, and the plasma membrane is just one of them. So amphipathic in the plasma membrane and amphipathic in digestion. And I thought you'd be interested, we can digress for just a bit here and think about soap. Soap is an amphipathic molecule, and here's how it works. So a soap molecule has a hydrophilic head and a hydrophobic tail. And if you introduce a droplet of oil into a solution containing soap, what forms is what's called a micelle. So all the tails go into the oil and the hydrophilic heads stick out into the water. So what is soap? How is soap made? Well, in the old days, people would take ashes plus lard and make soap. It's really that simple. Here I've got a couple of pictures of women making soap in big kettles. Here's an industrial factory for soap making where they're boiling ashes in these large vats. So to make soap is really very simple. You suspend ashes in water and boil it. Then you add fat and then add lye water, which is just the water that you get from cooking ashes. Add water to that until it gets thick, and then pour it into molds. As you see here, pour it into molds, and you can cut it up into bars of soap. So what actually happens, what is the chemistry of making soap? You start with a fat, and as you know, fats are triglycerides. That molecule is shown here. And if you add ashes, what do the ashes actually contribute? Sodium hydroxide. And so what you end up with is glycerol plus soap, in this case, sodium laurate. So you have the hydrophobic tails and the hydrophilic sodium ion. And here you see the structure of sodium laurate, a soap. So it's really very simple. Fat plus ashes gives you soap. When you wash your hands, what actually happens? In the lower right, you have the soap molecules in water. You have dirt on your skin. The hydrophobic tails bond to the dirt and form a micelle with the hydrophilic head sticking out into the water, and that comes loose from your skin and washes away. Some questions about soap or about hydrophilic, hydrophobic, amphipathic?
Oh, it's clear.
Yeah?
I'm saying it's clear. Thank you.
Okay. Okay, steroids and glycolipids. Cholesterol is found in plasma membranes, and it can either increase or decrease the membrane fluidity. You see the structure of cholesterol here. It's a nonpolar molecule, and it can be inserted at various places into the lipid, phospholipid bilayer. And that brings us to a consideration of the fluid mosaic model of a membrane. Now this is important, and so you really need to understand what this model is saying. Fluid is semi-solid, so the phospholipid layers are fluid. The membrane is a collage of proteins embedded in the matrix. So the membrane is not homogeneous. It's filled with what are called microdomains. So at this colored part of the picture is a drawing of a plasma membrane, the lipid bilayer, and then all these proteins and sugars embedded in it or on the surface. And here's a transmission electron micrograph of the same thing. So here's a statement of what the fluid mosaic membrane model is. The membrane components, which are proteins and glycolipids, form a mosaic of islands floating in a sea of phospholipids. So try to get that picture in your mind. At room temperature, the plasma membrane has the consistency of vegetable oil. So it's sort of in between a solid and a liquid. This allows the components of the membrane to float around in this viscous environment. And importantly, the membrane is not static, but rather it's in constant motion. So what are the factors that affect fluidity? Fluidity is the ability of a substance to flow. As fluidity increases, permeability increases. So remember that, it's in red. What are the factors that influence it? Well, temperature. As the temperature rises, fluidity increases. The fatty acids, the level of unsaturation promotes fluidity. In other words, the more unsaturated hydrocarbon chains there are, the more fluid the membrane will be. And finally, cholesterol, which acts, it's not a buffer, but it acts like a buffer. So at low temperature, when the membrane gets rigid, it increases fluidity. And at high temperature, when the membrane tends to fall apart, it decreases fluidity. So that's all illustrated in the diagram on the right. Membrane fluidity is affected by whether the hydrocarbons are saturated, in which it's rigid, or unsaturated, in which it's more flexible. And this graph shows the effect of temperature. At low temperature, membranes are solid, more solid-like. And as temperature rises, they become more fluid. And the role of cholesterol, as I said, is to disrupt the hydrophobic interactions and allow the membrane to adapt to the environment. So on the lower left are two important things to keep in mind. If the fluidity is too high, the membrane falls apart and the cell would die. If fluidity is too low, the membrane loses permeability, molecules cannot cross, and again, the cell would die. So this is the final slide I want to show you about membrane fluidity. And I hope you can get a vivid picture in your mind about what a membrane is actually like. The phospholipid bilayer is thick, viscous, semi-liquid. The proteins and glycolipids are embedded in the membrane, and they undergo continuous movement as though floating like a raft in a pond. So if you look at this picture and you see all these blue objects embedded in this red membrane, they're not immobile, they're not static, they're floating around and moving all the time. And the rate at which they move is a function of the fluidity of the membrane, whether the temperature is high or low, what the composition of the membrane is. I want to point out a couple of things. A peripheral membrane protein is shown here. This is attached to the membrane reversibly. In other words, it can come and go. And examples of this would be peptide hormones, antimicrobial peptides. If they're needed, they come in, attach to the membrane, do their job, and then they can leave. The other type of protein is called integral membrane protein. It's permanently attached to the membrane. You can see the integral proteins traverse the entire membrane from inside to outside. And these are the ones that are involved in transport of molecules or atoms into and out of the cell. So this is an important slide for you to keep in mind to understand what a plasma membrane is really like.
Excuse me, Professor.
Uh-huh.
So one of these protein might be for cystic fibrosis. That's for chloride.
Yes, so that would be an integral membrane protein. The CFTR protein would be it would be inserted through the entire membrane from inside to outside for chloride transport.
Yeah, so it might be in one cells, not like if it's needed. Like you said in the derma, it's near to the skin. So in these cells, we will find this gene there, this protein, right? It's not in all the cells, but in the cells that's in pancreas and lungs.
Yeah.
And we will find this protein there only.
It's also found in other types of cells and other organs, but the lungs and the pancreas are the main ones that are affected by that disease, by that mutation.
Okay.
Another quick question, if we could.
Yeah.
So I'm seeing the peripheral proteins only on the inside of the bilayer. Is there any situation in which you'd have them on the outside of the bilayer, or is it strictly an interior thing?
The only place I've ever seen them located is on the interior.
Okay.
Scott, do you have any more information on that?
Okay, you are hearing me. They can be external. I can't. I have heard that they can be external, but I could not cite you a source or a particular type of protein. I also tend to only think of peripheral proteins as being on the inside of the plasma membrane.
Okay. Sometimes maybe. Got it.
Okay, anything else?
Yeah, one quick question. So in cystic fibrosis, what's the problem? It's that we don't have these genes in the gene in the membrane. So that's what lock the way for the chloride. So it doesn't run. That's what make problem. Yeah, in cystic fibrosis, we don't have this protein in the membrane, right?
You may or may not. Remember, there are a lot of different mutations in the CFTR gene. And those mutations can block the protein at any number of different points as it goes from the nucleus through the cytoplasm and ends up in the membrane. So some cystic fibrosis mutations allow a protein to be formed. It gets into the membrane, but it doesn't function efficiently. It transports chloride ions poorly. Other times, the protein is not made at all. Other mutations cause it to abort, in which case there is no protein in the membrane at all.
Oh yeah. So if it wasn't there, it's one case of a mutation.
Yeah, one type of mutation
Yes.
would, in fact, I think two types of mutation end up with no protein at all in the membrane. The other four types of mutation, you have CFTR protein in the membrane, but it doesn't function properly.
Oh, thank you. I get that.
Yeah, that's an important point. And your cystic fibrosis project will deal directly with this issue. Okay, we're switching now from consideration of the physical characteristics of the plasma membrane to a consideration of osmosis and diffusion. First, what is the definition of diffusion? It's a movement of molecules from higher concentration to lower concentration. And it's based on kinetic energy of the molecules. You've all done what's shown here in the pictures on the right. You put a drop of food coloring in a glass of water. And at first, there's this red blob where the drop landed. But as you wait, that drop disperses. And finally, if you go all the way to the right here, the food coloring disperses evenly throughout the water. And that's called diffusion. It's the passive movement of molecules from high concentration to low. What then is osmosis? It's diffusion through a semi-permeable membrane. And I've circled that, so make sure you know the definition of osmosis. It's the movement of a solvent, and that's in a biological system, that would be water, through a semi-permeable membrane into a solution of higher solute concentration. So what is a solvent? It's a liquid that can dissolve other substances. A solute is a substance that can be dissolved. And we'll talk now next about tonicity of solutions, isotonic, hypotonic, and hypertonic. But first, look at the pictures on the right. We have two beakers separated by a semi-permeable membrane. And in this case, this membrane is permeable to water, but not to salt. So when osmosis occurs through the tube connecting the two beakers, and notice what happens to the water level.
The water level changed because of the hypertonic. It shows hypertonic and hypotonic also, teacher.
Well, it shows that the water level in the beaker on the right goes up. And why does it go up? Because of osmosis. The water is moving into the beaker that has the solute. So if you remember how solutions form, when we talked about sodium chloride, and that's what's being used here, water molecules hydrogen bond to both the chloride ions and the sodium ions. And that's what puts them in solution. Water, osmosis, is diffusion through a semi-permeable membrane because it can go through the membrane, but the salt molecules cannot. So they stay there, and osmosis causes the level of the water to rise as a result. Okay. Okay, tonicity of solutions. In an isotonic environment, the water concentration outside the cell is equal to the water concentration inside. So that's easy to understand. However, most cells contain about 0.9% dissolved salts. That's about the concentration of salts in seawater. So an isotonic environment must contain about 0.9% salts. If you want a cell to be happy, since a cell has 0.9% dissolved salts inside, it needs 0.9% outside as well. What is hypotonic? And we're going to talk about hypotonic with respect to the environment of the cell, what kind of an environment the cell is in. The water concentration outside the cell is higher than inside, or to look at it the other way, solute concentration outside is lower than inside. And as a result, water moves into a cell at a greater rate than it moves out. A solution with the lowest concentration of solute is described as hypotonic. In contrast to that, hypertonic, and again, this is with respect to the environment of the cell. The water concentration outside the cell is lower than inside. And as a result, the water moves out of the cell at a greater rate than it moves in. The solution with the highest concentration of solute is described as hypertonic. And this slide illustrates that. Circled here in red, hypotonic, hypo means below, means cell water is low, solutes in the cell are high, and water flows into the cell, and the cell is turgid. It has a lot of water in it. Hypertonic, hyper means above, means cell water is high, solutes are low, water flows out of the cell, and that causes a condition known as plasmolysis. And you see that these red blood cells here, in a hypertonic solution, the cells shrivel as water moves out of them. Isotonic, they're normal, and hypotonic, water flows in and they swell up and can even burst if the osmotic pressure is high enough. Same things are illustrated here to the right comparing both plant and animal cells. In a hypertonic solution, the cell will shrivel, whether it's an animal cell or a plant cell. Remember, a plant cell has a rigid cell wall, so the cell shrivels inside that wall. Whereas in a hypotonic solution, water flows into the cell, and the cell is very turgid. In the case of an animal cell, the cell could burst. Scott, you have a particular way of describing these things. Would you like to comment? Scott, are you there? Okay, then we will go on. I put this in just to remind you of the vacuole in plant cells. Remember, plant cells have a large vacuole, and one of the main functions of this large vacuole that animal cells don't have is to store water. So under hypotonic conditions, the vacuole takes in a lot of water, the cell is turgid, whereas under hypertonic conditions, the water flows out of the cell and the cytoplasm is all shrunk up inside the cell wall.
Professor, I have a quick question.
Uh-huh.
For plant cells, would it be better for them to be in isotonic or hypotonic situations?
Oh, it's better for them to be isotonic, where the environment that they're in is has an equal osmotic pressure to the environment inside the cell. And so for all types of cells, that's the equivalent of 0.9% salts.
Gotcha. Okay. Thank you.
Okay. See if Scott, are you back yet?
Yes, sir, I am.
Okay, I just wondered if you wanted to make a comment about these definitions, hypo and hyper, and about the workshop that you have on these.
So you really need to know the definitions because people get very confused on this. It's not terribly difficult once you've played with it a little bit, but until you have, your best defense is know what these definitions mean. I do cover this usually. I will spend a couple hours in workshops talking about this and giving examples for people because again, it's not hard, but you got to play with it a little bit. And I have a process that I like to use that makes it relatively goof proof to answer these questions. So if this isn't making a whole bunch of sense to you, definitely consider coming to the workshop.
And you do have a worksheet that has several questions about hypo and hypertonic situations.
Excuse me, teacher, I have a question regarding to this isotonic and hypotonic and hypertonic. These situations are related to hydration of plants or human body. When a person good hydrated, it's maybe the isotonic condition is occurs, or when a person go through the loss a lot of water or dehydration, it goes to the cells go to hypotonic condition.
Yeah, if cells are dehydrated, that they would be in a hypertonic condition.
Thank you, teacher.
Okay. We're going to talk about movement of molecules across the membrane. There are three possible types of transport. Molecules can actually diffuse passively across the membrane. There's passive transport, also known as facilitated diffusion. There's no energy is required for that. And then there's active transport, which requires an expenditure of energy. So what you see here is a rough approximation of each of these. Diffusion just occurs across the membrane from high concentration to low without any expenditure of energy. Passive transport occurs through a channel, but it is passive, does not require energy. Whereas active transport occurs, and this is important, against the concentration gradient. So you can see that the concentration of the blue balls is higher outside the cell, lower inside the cell, and yet transport is going from lower to higher concentration, and that requires the cell to expend ATP to power the movement against a concentration gradient. Okay, passive transport proteins. No energy is required. The channels are open or they can be gated. And the overall movement is from high concentration to low. So if you look at the two types of channel, it can be an open channel or it can be a gated channel. But in both cases, if you look at, say, the bright green dots, they're at a high concentration outside the cell, and they're moving into the interior of the cell where they're at a lower concentration. And the same thing is true for the larger pale green objects passing through a gated channel. They go from high concentration to low. Now, in contrast to that, there is active transport. It requires that a cell expends energy to move molecules across the membrane. A transport protein actively pumps a specific solute against that solute's concentration gradient. Systems like this are common, coupling the passage of two solutes can transport ions as well as uncharged molecules, and the source of energy is usually ATP. So the functions, a cell can take up molecules against a concentration gradient. It can also move molecules out of the cell against a concentration gradient so that the cell can maintain an optimal concentration of ions. And again, I'm emphasizing that the transport protein usually uses ATP. It can be other sources of energy, but it's usually ATP because you're going against the concentration gradient. A classical example of active transport is the sodium-potassium pump, which transports sodium out of the cell and transports potassium in. So you see here on the left-hand side, sodium ions entering the transport protein, the pump, which then uses ATP to open on the outside and release the sodium ions outside the cell. And conversely, potassium ions can enter the cell from the outside, and when phosphate is hydrolyzed to release energy, they can be released to the inside of the cell. So in both cases, the ion is moving against its own concentration gradient. There are several points to be made on this slide about the sodium-potassium pump. There are several points. And one final point I want to make is that there are there are electrochemical gradients. In fact, most of the time, the gradients are electrochemical. The chemical gradient is the differential between concentrations of substances inside and outside. So in this case, sodium concentration inside is high, concentration outside is low, so that is a chemical gradient. The electrical gradient is because, as a result of that, the outside of the cell has a positive charge, the inside of the cell has a negative charge. So both phenomena are going on at the same time. If the concentration of the chemical is different, the electrical charge is also in a gradient across the membrane. And finally, getting back to CFTR, it's a gated channel protein that requires ATP. And we talked about this a little earlier, but chloride ions need to be transported across the membrane, and they do so by if the CFTR protein utilizes ATP to generate energy. The point here is that sometimes instead of adenosine, guanosine triphosphate is used as the energy source. And now finally, let's talk about transport across membranes of large molecules. there are two types of process. One is called exocytosis, which exports molecules from the cell. Endocytosis brings molecules in. And this picture illustrates both of those. Endocytosis, molecules get concentrated in coated pits in the plasma membrane, which pinch off to form vesicles. And these vesicles then can deliver the contents to various places in the cell. And similarly, or I should say conversely, if the cell is trying to get rid of substances, they can be packaged into a vesicle, which is exported to the plasma membrane where it fuses and releases the unwanted material to the outside of the cell. So endocytosis and exocytosis. This focuses on exocytosis. Here's an electron micrograph of exocytosis underway. There's a transport vesicle. It fuses to the membrane and releases the unwanted substance, in this case, a protein. If we look at endocytosis, that's taking things into the cell. There are three types: pinocytosis, receptor-mediated endocytosis, and phagocytosis. So pinocytosis is the uptake of an extracellular fluid. It forms a pit in the membrane and pinches off as a vesicle. Receptor-mediated, this is CFTR, as an example, where the plasma membrane has a receptor. The virus, or excuse me, receptor-mediated, not CFTR. This is coronavirus as an example, where the ACE2 protein is on the membrane. The virus has a spike, and the spike binds to the receptor, and a membrane deformation occurs, forms a coated pit, and the virus gets taken into the cell inside a vesicle. A phagocytosis is for very large substances, like a bacterial cell, but the same process occurs. The plasma membrane invaginates, pinches off, and brings in this case, a bacterial cell into the host cell. Now I've put at the end of this slide set a set of summary points. If you want to read about this in the textbook, the pages are given there, 63 to 89. So you can, in your study, you can consider these points that I've made with respect to all the information on the preceding slide set. And this slide set is in your Canvas container, as will be the recording of this session. Okay, our time is up. Any questions or comments or thoughts before we end the session? Scott, would you like to say something about workshop today?
Pretty much I've said it. Workshop is going to be in about 20 minutes, and primarily history shows that people like to spend some time learning about tonicity and osmosis stuff. But feel free to bring it all, and we can discuss things.
Yeah, and I'd strongly encourage you to go see Scott about tonicity and membrane functions. It's complicated, not always completely intuitive, and Scott can help you out with that.
When is our next quiz?
Next quiz is a week from today, the 22nd.
And what would the topics be about?
Well, the topics will be what we haven't covered since the last quiz. So that would include cystic fibrosis, coronavirus, and today's lecture, membranes.
Okay, thank you.
I was checking the PowerPoint for today and it says it's still not published.
Oh, really?
Yeah.
Ah, okay. Well, thank you for telling me that. Okay, I'll make sure that it gets published.
Thank you.
Okay, anything else? All right, then I'm going to stop sharing, and I'll see you all on Wednesday. I'm going to end the
Have a
session.
good one.
Okay, bye.
Have a good day.